Hello, hi everyone. So today I would like to tell you the part two of Sanger sequencing and the NGS difference. So let's have a look at the benefit and the challenges between the two sequencing techniques. So let's have a look at the benefit of the Sanger sequence. It is very fast and it is cost-effective sequencing. Especially, we can use Sanger sequencing for a lower number of targets. If the target is on average from 1 to 20 targets, so in that case, we better use Sanger sequencing and the familiar workflow. Whereas the benefit of the targeted NGS, the next generation sequencing, as we have said already in the previous lesson, it is a higher sequencing depth that can enable with higher sensitivity. So it has high sensitivity. So the other part is it has higher discovery power. So when we say discovery, its ability to identify the novel variant. So in case if there is a novel variant, with NGS, we can easily identify that variant. So it is very high. And the other one is higher mutation resolution, and that's also another benefit. Uh, and the other part is the more data produced with the same amount of input in the DNA and higher sample throughputs. So what could be the challenge? What could be the challenge of Sanger sequencing? Obviously, it has low sensitivity. Whereas in case of NGS, we say that already. It is higher sensitivity there with higher sequencing depth. So one of the limitations with Sanger sequencing is it is low sensitivity and also low discovery power. In case if we look a novel variants. So in that case, the ability of the Sanger sequencing to identify that novel variant is very low. So that's also another, and not as cost effective for a high number of targets. For example, if it is greater than 20, it's not as cost effective, right? And low scalability due to increasing sample input requirements. So these are one of the limitation. And regarding with NGS, less cost effective for sequencing low number of targets. So we cannot sequence. Yeah, it is cost effective. It will be very high. If we go to sequence with NGS with a target that is from 1 to 20, and it's really time consuming for sequencing for a very low number of targets. So if we have definitely a lower targets, we prefer Sanger sequencing, and if we have higher targets which could be greater than 20, and in that case we better use NGS, the next generation of sequencing. So these are the two main differences between the Sanger and the NGS. So what could be the advantage, the benefit of NGS, or the advantage of NGS versus the Sanger sequencing? As we have said before, it has higher sensitivity to detect a low frequency variant. So maybe in a sample we may have low frequency variant. So with that variant, it doesn't matter whether it is low, very little. So with NGS we can detect that low frequency variants. So it has a high sensitivity and faster turnaround time for high sample volume and comprehensive genomic coverage, and lower limit of detection, and higher throughput with sample multiplexing. And as we have said already, the ability to sequence hundreds to thousands of genes or the gene region simultaneously. So therefore, with Sanger sequencing, we saw a limited DNA snapshot. Whereas with NGS and its massively parallel sequencing enables us to look at tens to hundreds of thousands of read per sample. So that is, the main difference with the main benefit of NGS with the Sanger sequencing. I hope you got something from this video, and in case if you need to to see further about the differences about NGS and Sanger sequence, just go to the link below and read. And if you like another video, just share it and like it and subscribe. And I wish I'll bring another video. I'll come in with another video with more application of the Sanger and the NGS in science. So have a nice time.